Head and neck squamous cell carcinoma, or HNSCC, typically does not respond well to immunotherapy. A key characteristic of HNSCC is that the majority of cases carry TP53 mutations. The absence of functional p53, a key protein for tumor suppression, alters the tumor microenvironment, helping HNSCC tumors evade immune responses. Recent research has identified the autonomic nervous system, specifically beta-adrenergic signaling, as an important regulator of cancer progression. Beta-adrenergic receptors, or beta-2ARs, are found on both cancer and immune cells, and their activation generally suppresses immune cells' ability to fight tumors. However, the interplay between beta-adrenergic signaling and p53 loss remains poorly understood. To address this, researchers from the University of Texas investigated the effects of beta-2AR activation on the immune sensitivity of TP53 mutant HNSCC tumors. In vitro experiments showed that beta-2ARs are highly expressed in oral cancer cells. Pharmacologically activating these receptors with isoprenaline altered the secretory profile of these cells, specifically triggering the release of cytokines known to activate cytotoxic T cells, especially CXCL10. Interestingly, beta-2AR activation did not produce the same effect in tumors with functional TP53. Cytotoxic T cells became activated upon exposure to conditioned medium from beta-2AR activated p53 deficient tumor cells, upregulating genes associated with cytotoxic activity, including those induced by the activation of CXCR3, TNF, and IL-6 receptors. Activation failed when anti-CXCL10 antibodies were added to the medium, confirming that CXCL10 was crucial for T-cell activation. Prolonged CXCL10 exposure drove these T-cells toward an exhaustion-like state. However, the T-cells continued to express cytotoxic proteins, which indicates a non-traditional state of exhaustion with latent cytotoxic potential. In vivo tests in Th-Flox mouse model with impaired adrenergic signaling showed that restoring beta-2AR activity in the p53 deficient tumor microenvironment greatly increased CXCL10 secretion and infiltration of the tumor by cytotoxic T-cells. These findings challenge the previously held belief that beta-adrenergic signaling suppresses anti-tumor immunity, demonstrating instead that in TP53 null HNSCC, beta-2-adrenergic stimulation enhances cytotoxic T-cell activity. Collectively, these findings uncover a neuroimmune axis that reverses immune escape in p53 deficient HNSCC and point to novel therapeutic strategies targeting adrenergic signaling to convert immune cold tumors into immune hot ones more amenable to immunotherapy.